begin our lecture on carbohydrates today. We will have two lectures on carbohydrates, and we will see how important they are in the processes that go on in the body later on, especially when we do bioenergetics, where we will be studying the metabolism of these carbohydrates. Now, the first thing that we want to know is what are carbohydrates. Carbohydrates are actually aldehyde or ketone compounds with multiple hydroxyl groups, okay? And they have a lot of functions in our body, and the main functions are, the first one, which is most important, is as energy stores in the body, as fuel and metabolic intermediates that give us our basic energetics, that the energy that we need actually to get along. The next important thing is that they form the part of the structural framework of RNA and DNA, the essential molecules for life. Remember, when we consider the central dogma of biology, it was to go from DNA to RNA to protein. Okay? Now, these carbohydrates actually form the basic structural framework of RNA and DNA, and when we do the structures of nucleic acids and their components, we will see how this actually comes into the picture. They also form the structural elements in the cell walls of bacteria and plants, and in the exoskeleton of arthropods. Okay? This is what forms the chitin, which you have probably heard about, the exoskeleton of these arthropods like cockroaches, and it is that hard shell that is formed. That is also a polysaccharide, which also is basically a carbohydrate. They are also linked to many proteins and lipids, and when we studied lipids and the fluid mosaic membrane of lipids, we found out there were proteins which we called glycoproteins that had sugar attached to the lipid moieties that were part of the fluid mosaic membrane that allowed the transfer of material from the inside to the outside of the cell. They also play a key role in cell-cell recognition processes. So, these are the major functions of these carbohydrates that are basically aldehyde or ketone compounds that have multiple hydroxyl groups. Now, basically these polyhydroxy alcohols, as you probably might all know, they have the general formula CH2O, the whole N. And what is this N? It is the number of carbon atoms that are present. For example, glycerol, which was something we also used in the lipids, right? In this, here also glycerol can be considered to be the parent compound, even though itself it is not a carbohydrate. What can happen to this glycerol is it can be oxidized. It can be oxidized to form an aldehyde or a ketone, and these actually form the basis for all carbohydrates. So, if we just go to look at the structure of glycerol, this is what glycerol is: CH2OH, CH2OH, CH2OH. So, this is glycerol, and when we studied the fatty acids and the lipids, we found out that these formed actually, they were linked to fatty acids forming an ester, and then we had a phosphate linkage and then some extra linkages here that actually gave us the polar head group and the two hydrophobic tails of our lipid molecule. Now, when we come to carbohydrates again, we see that glycerol again here is the parent compound. What is happening here is there is oxidation. Oxidation to form glyceraldehyde, this is CHOH. Okay. So, this is actually glycerol. So, we have glycerol here, and we have, what is this? This is glyceraldehyde. We can also form CH2OH, C double bond O, CH2OH, which is called dihydroxyacetone. Dihydroxyacetone. acetone. Okay. So, we know that acetone is CH3COCH3. This is dihydroxyacetone. So, we have again the parent compound being glycerol, and from this we can get these two aldehyde and ketone components that actually form the basis for all carbohydrates. So, we have carbohydrates that contain an aldehyde like glyceraldehyde that are called aldoses, and glyceraldehyde is an aldotriose because it has 3 carbon atoms, an aldose with 3 carbon atoms. We have ketoses like the one I just drew, dihydroxyacetone, where the carbohydrate contains a ketone. It is a ketotriose, so it is a ketose with 3 carbon atoms. Okay, this is just the nomenclature that you would look at. So, you have an aldose, in this case glyceraldehyde is an aldotriose. We have ketoses, dihydroxyacetone is a ketotriose. Now, these are the structures that we just looked at. We have our glyceraldehyde and aldotriose, and we have dihydroxyacetone, a ketotriose. Okay. Now, if we look at the carbon atoms here, this carbon atom, the one that's in the center here, is an asymmetric carbon atom, right? Why? Because it has 4 different groups attached to it. We can form therefore an isomer of glyceraldehyde if the OH is on the other side. If the OH is on the left, the OH is on the right here. If the OH is on the left, then we form an isomer. But that is not true in the case of the ketotriose. We do not have a chiral center here. Because we have identical groups, there is no carbon atom that has 4 different groups attached to it. Okay? So, when we look at aldoses and ketoses, the number of isomers are different depending on the number of carbon atoms present. So, if we have 3 carbon atoms like in this aldotriose, there is a possibility of 2 isomers being formed because this OH can flip from the right to the left. Whereas, for the ketotriose, this is the only isomer that is possible. Okay? So, depending on the number of, then when we have an aldotetrose, we have 4 carbon atoms and we have 4 isomers possible. Okay? Similarly, when we have a ketotetrose, what does that mean? A ketone with 4 carbon atoms in that case, you could have a possibility of 2 isomers depending on the number of chiral carbons that you would have and where you could place the OH in 3D structure. So, if we look at glyceraldehyde once more, we have these 4 different groups attached to it. 4 different groups attached to it means that there are 2 such stereoisomers of glyceraldehyde possible. One is D-glyceraldehyde and the other is L-glyceraldehyde. Okay? The L-glyceraldehyde, the OH is on the left. In D-glyceraldehyde, the OH group is on the right. So, this is the stereochemistry that is followed by convention for all carbohydrates. By convention, the sugars are written in such a manner that the most oxidized carbon, that's the aldehyde or the ketone, is written to the top of the structure, at the top. The chiral center that is farthest from the oxidized group is the one that decides whether it's D or L, not any other OH group. So, what we have is we have the sugars written with the most oxidized carbon, that is the aldehyde or the ketone at the top, and the chiral center that's farthest from the oxidized carbon will determine whether it's D or L. If the hydroxyl group is to the left, it is the L configuration. If it is to the right, it is the D configuration, and in general use, there are D isomers in biological compounds mostly. Okay. So, we have some other aldoses that we can consider here. So, this is 3 carbon atoms, we have D-glyceraldehyde. When we look at 4 carbons, we have D-erythrose. Why is this D? The most oxidized carbon is at the top. The carbon atom that is chiral, that is furthest away from the oxidized carbon, has the OH to the right, which means this is D-erythrose. This is also D3-ose because the OH attached to the chiral carbon farthest away from the oxidized carbon is on the right. Okay? So, that is your nomenclature. In D-ketoses, we have what we have looked at before. With 3 carbons, you have dihydroxyacetone. When we are looking at 4 carbons, again we have something, a carbohydrate called erythrulose, where we have, what is this? This is a ketotetrose. It has 4 carbon atoms and it is a ketose. We have now the oxidized carbon as further to the top as possible. We have the chiral carbon with the OH to the right. So, it is a D-erythrulose. We can also have other types of D-aldoses, where when we look at the 6 carbons that we are going to consider, we have allose, altrose, glucose, mannose, gulose, idose, galactose and talose. Okay? But what we need to know is what is important for most biological carbohydrates is glucose, mannose and galactose. Okay? Now, when we have this, so what do we see? We see all of these are actually D structures that have been drawn here. So, the most oxidized carbon is at the top. The farthest chiral center away from the oxidized carbon has the OH on the right. So, this is a D-glucose. The glucose one is the one that we are going to be most interested in, and we will look at this structure in much more detail as we go along. These are other sugars that these structures are important now for all of the sugars that we are going to do later. Glucose is an aldohexose. Why? Because it has an aldehyde group and it has 6 carbon atoms. So, it is an aldohexose. Fructose is a ketohexose. It has 6 carbon atoms and it has a ketone group to it. Ribose, which forms the basis of ribonucleic acid, the basic sugar in ribonucleic acid, is an aldopentose. And the sugar that forms the basis for all DNA molecules is 2-deoxyribose, where the numbering begins from always the most oxidized carbon atom. So, this is number 1, this is number 2. The deoxy means it has lost this. So, it is now CH2. So, this is deoxyribose. So, what we have for ribose that is going to form the basis for ribonucleic acid is the ribose sugar, which is an aldopentose. For deoxyribonucleic acid, the basis is 2-deoxyribose, which is missing an OH, and we will see how important this is in a lot of structures. For example, when we did the mechanism of ribonuclease, we found out that the ribonuclease, because it acts on this 2' OH, could not act on DNA and cleave DNA. It could only cleave ribonucleic acid, if you remember when we did enzyme mechanisms. So, these are the small biomolecules that are extremely important, or probably the most important ones, where we have glucose, fructose, ribose and 2-deoxyribose. So, these are some other important monosaccharides. D-glyceraldehyde is the simplest of sugars. D-glucose is the most important one in the diet. We will see how this is also stored and how it is even broken down in the body. Then we have D-fructose, which happens to be the sweetest of all sugars. We have D-galactose that forms a part of milk sugar, and as I said, D-ribose that is used in RNA, and D-deoxyribose, 2-deoxyribose that is used in DNA, and all of these you see are D enantiomers. And when we looked at proteins, we found that most of them were L amino acids. Okay. Now, this is what we mean by something called epimers. First of all, let us look at the numbering of glucose. If we look at the numbering of glucose, we find that what is glucose basically? It is an aldohexose. Okay. Now, the aldohexose has its aldehyde group on the top. This is the way it is written, and the rest of the molecule follows. The chiral carbon, that is number 5 here, which is the furthest away from the oxidized carbon, will determine whether it is D or L. This is on the right, so it is D glucose. We have now 2 other aldohexoses here, D-mannose and D-galactose. Okay. Now, what we have here is, if you look at the structures carefully, in this case the change in the orientation of the OH is on carbon number 2. The rest is the same. We have the OH on the left in mannose and on the right in glucose. However, for carbon numbers 3, 4 and 5, the OH directions are exactly the same. The only change that you see is in carbon number 2 between D-glucose and D-mannose. So, what you say is, when you have the difference in the stereochemistry at only one carbon atom, it is referred to as what is called an epimer. So, this is an epimer at C2. So, you would call this an epimer at C2, meaning that at carbon number 2, this would be an epimer of D-glucose at carbon number 2. So, since we all have to remember the structure of D-glucose, and if it were to be mentioned that you know the structure of D-glucose and you know that you have D-mannose, which is an epimer of D-glucose at carbon number 2, you would know that the stereochemistry at carbon number 2 is the only one that is different. So, instead of having the OH on the right, you would write the OH on the left. If I say now that there is another epimer of glucose, that is an epimer at C4, that is called D-galactose. So, now since we know the structure of glucose, I know that the only stereochemistry, or the carbon that is going to change the stereochemistry, is going to be at carbon number 4. So, in carbon number 4, instead of having the OH on the right, I am going to have the OH on the left. So, that would be the epimer at C4, which is D-galactose. Okay, so, all we would have to remember actually is the structure of glucose, and then know that D-galactose is an epimer of glucose at C4 and D-mannose is an epimer of glucose at C2. Okay. So, that would be what we would look at. Now, we look at the other structures. Okay. So, let us look at now other structures of glucose. We have what is called hemiacetal formation. This is something that you probably studied before also, but nevertheless we will go through it, because it is going to be important in the understanding of what goes on later on in terms of its breakage and cleavage. We have an aldehyde. An aldehyde reacts with an alcohol. Now, when it reacts with this alcohol, then it can form what is called an hemiacetal. What is this hemiacetal? It has an OH and it has an OR2. Now, what is attached here is the rest of the chain. We have the aldehyde group and we have an alcohol group here. What happens here is this OR2, the lone pair on the oxygen, is going to attack this carbon. In doing so, it attacks this carbon. So, what happens here? This goes and takes up this proton. So, it forms, now you see this proton is marked in red, which has actually come from the alcohol, and the OR2 is now linked to the carbon to form what is called an hemiacetal. So, we can also form an acetal. What is going to happen to the acetal now? We are going to have again this attack the carbon here, and what is going to be released now? The OH. The OH is going to take up this H and form H2O and an acetal. Okay. So, this is extremely important in the cyclization of glucose, which we will see in a moment, and we have two such forms of glucose called an alpha form and a beta form. Basically, what is happening is, in the glucose structure we have not only an aldehyde group, but we also have an alcohol group. So, what does it actually look like? We have here not only an aldehyde group, so we have C, this is what we have. So, if we write this actually in a different fashion, let us just write it out straight first. This is just an example where we are showing how we can actually have. So, where is our aldehyde? Our aldehyde is sitting here, and where is our alcohol? It is here. So, what can actually happen? I can have cyclization, right. So, if we write this in a manner that is going to be like this, actually it is not this one, it is this one that actually reacts, this alcohol. So, we have OH, CH2OH, C, OH. This will be apparent when I show you the slide also, but you should draw it yourselves once. Okay, so we have an H also sticking here. So, if we just number the carbons, let us be careful in our numbering. We have this as carbon number 1, 2, 3, 4, 5 and 6, right. When we look at the carbons here, this is 1, 2, 3, 4, 5 and 6. So, which is the alcohol that is actually forming the hemiacetal? It is this alcohol with this aldehyde, right, because this CH2OH is up there, number 6. So, the numbering is 1, 2, 3, 4, 5 and 6. So, now if we have the same reaction going on, what is going to happen is this from the oxygen is going to come here, and what is this going to do? It is going to take up this proton, and what are we going to form? We are going to form and CH2OH. Fine. The rest are all the hydrogens. So, that is what we formed. Now, so what have we actually done? If we go back to the hemiacetal formation here, we have the aldehyde with the alcohol forming an hemiacetal. Okay. Now, if we look at this is what is happening. We have the cyclization where it is this OH attached to 5 that is doing what? Participating with the aldehyde. So, the hydroxyl oxygen and the aldehyde of carbon number 1 is going to do what? Cyclize, and what will we have? We will get a 6-membered ring here. One of the members is going to be the oxygen, a 6-membered ring with the CH2OH being the 6th carbon. We can also have 5-membered rings, right? Because we can also have the cyclization of D-fructose. Okay? We will look at what those rings look like in a moment. So, since these 6-membered rings resemble pyran, they are called pyranosides. So, glucose is also called glucopyranoside. Okay? Furanoside are 5-membered rings with oxygens because they resemble furan. Okay? And we usually have them drawn as Haworth projections as was shown before. Okay, so what do we have? We have 6-membered rings and 5-membered rings that resemble pyran and furan. So, they are also called pyranosides and furanosides. So, this is what we have. You should try and work out the structure for the case of furanose and see what it looks like. Okay? You should get a 5-membered ring in that case because we have the C double bond O, but to the C double bond O we have a CH2OH attached, right? So, now if we look at the alpha D glucopyranose, now we will see what this alpha is in a moment. But for now we have to know that we have a 6-membered ring here and we have the oxygen resembling the structure of a pyran. So, we have glucopyranose. The 5-membered ring in case of fructose, which is a ketohexose, is forming a fructofuranose. Now, there are some things that we can notice here, especially at carbon atom number 1. Carbon atom number 1, in the first diagram we have the OH that is trans to the CH2OH. In the second diagram we have the OH cis to the CH2OH, which is the 6th carbon. So, what do we have? We have what is called an alpha anomer and a beta anomer, and you form anomers of glucose only when you have the closed ring structure. So, what do we have? We have in this case the OH on carbon atom number 1 that we have to take care of. If this OH is trans to the CH2OH, it is called an alpha anomer. If it is cis to the CH2OH, it is known as a beta anomer. The same goes with fructofuranose. The CH2OH, if it is cis to the other CH2OH, it is the alpha. If it is trans, then it is the beta form. Okay? This is what are the anomers. So, the different stereoisomers alpha and beta are called anomers, and we have the aldehyde or ketone carbon, which will give us or decide whether we have the anomeric in the beta position or in the alpha position, and you have to remember that this is only possible when there is cyclization, not before that. So, we have the alpha OH down compared to CH2OH, that is trans. If the beta OH group is up compared to CH2OH, it is basically a cis conformation when it is beta, and we have the alpha, and we will see how this is going to help us in all the glycosidic linkages that we talk about later on. Okay, so this is what we have. We have our glucose here, we have the cyclization, and what is the cyclization leading us to? When we have the OH trans to the CH2OH, we have alpha D glucose. When we have the OH cis to CH2OH, it is beta D glucose. When we have the ketose sugar, what do we have? In this case, they have shown the OH trans to the CH2OH and the OH cis to the CH2OH. Now conformations. We have 6 membered rings for our glucose, which means that we can have either a boat conformation or a chair conformation. This is something you already know with 6 membered rings. And what do we have is we can have axial bonds or equatorial bonds depending on the disposition of why is this shaped like this? Because this carbon atom is sp3 hybridized, so it has a tetrahedral disposition conformation to it, which means that you can have an axial part and an equatorial part. And we also know that the chair conformation is favored over the boat conformation, because in this case we have steric crowding. Okay. Now of these 2 major conformations that are available, so this would be the structure of glucose. We have the 6 membered ring, which is our pyranose ring, because we have the oxygen sitting here, we have the OH, and we have the OH in this case it is in the equatorial form, here it is in the axial form. So we have a stable chair conformation because it is obviously going to reduce the steric clash between the ring substituents. This is something you all know already. So we have here the axial and the equatorial axis, and there are 2 possible chair conformations where we have the oxygen here or here. What we can have here, this is alpha D glucopyranose. Now you have to look at the name carefully. Pyranose means it has a 6 membered ring with oxygen in it. We have the OH trans to the CH2OH, right? So it is alpha. And why is it D? It is D because the last chiral carbon atom away from the most oxidized carbon atom is having the OH on the right. So it is alpha D glucopyranose. Another thing that we have to consider is in aqueous solution the different anomers can actually interconvert between themselves. Now this interconversion actually gives us a transient formation of the open chain form, right? Because you realize that once it goes through the reaction, there is going to be a certain intermediate that is going to have the alpha chain in the open conformation. Now when it has it in the open conformation, when it closes again, the OH could either be up or down, that is cis or trans to the CH2OH that is on the 6th carbon atom. So what can happen is in aqueous solution, when we have the different anomers that can actually interconvert due to the transient formation of the open chain form, this process is known as Mutter rotation. Okay? And the ratio of the 2 forms is actually determined by their relative stability, and in the case of D-glucose, the beta anomer is more stable because it results in the C1 hydroxyl group in the equatorial position. You know that when we have the chair conformation, first of all between the boat and the chair, we know that the chair conformation is more stable. Now again, when we are looking at the chair conformation, if we want to look at which of the chair conformations are stable, we know that we have to look at the chair conformation which has the most bulkier substituents at the equatorial positions. So that is exactly in the case of the beta anomer, where the hydroxyl group is in the equatorial position, and that will make it the stable form for D-glucose. So this is what we have in looking at the structures of our glucose. We have basically what are we forming here? We have our aldehyde group, we have the OH that is attached to the carbon number 5. Okay? We have the formation, or we basically have our cyclization. In this cyclization, it is possible that the OH either goes trans to the CH2OH or it goes cis to the CH2OH. We usually use, or we refer to these as pyranoses because it is based on pyran. And then we also know that when we look at the chair conformation of this, it would be more convenient to have the OH group at an equatorial position, so that there would be more, or basically there will not be any bulky groups at actual positions, which would be unfavorable. Okay. Now let us look at the reactions of the glucose and other monosaccharides. There are certain reactions of glucose that are important for the metabolic breakdown of glucose. Okay. There is oxidation and reduction that can occur. Oxidation and reduction required for the complete metabolic breakdown of glucose. This is extremely important. And another thing important about glucose is storage. It is not stored as glucose, because glucose breaks down. Okay? There is a metabolic breakdown of glucose. So, when it is stored as glycogen. Okay? We will see in the next class what the different types of methodologies are, or the different types of linkages are, and how we can actually store them. Okay, so we have our different reactions. One is oxidation reduction. What is this oxidation reduction? We require this, we will see later on when we do bioenergetics and consider the metabolism of carbohydrates and how this is important there. We have esterification, where we have the production of phosphate esters. We have amino derivatives, where we have the use to produce structural components and glycoproteins. What are these glycoproteins? These glycoproteins are those that have glucose linked to the protein. Okay? And we will see how they are actually linked to the protein. Then the most important thing that we will be considering for disaccharide formation or polysaccharide formation is glycosidic linkages. Okay? So, when we consider glycoside formation, we will understand what these linkages actually are in their formation. For example, when we consider, we know that we have an alpha form, we know that we have a beta form. Okay? Now, if we just look at the structure of glucose in the different forms, then we have OH, CH2OH, H, what is this form? Okay. Now, what is happening here? Why is it the beta form? Because the OH is cis to the CH2OH. So, it is beta. Okay. Now, when we talk of these linkages, okay, we have to first of all remember the numbering of the carbon. Okay. What is the number of this carbon? It is number 1. This is number 2, this is number 3, 4, 5 and 6. What happened here? We had a hemiacetal formation. Why? Because we had the aldehyde and the OH of carbon number 5 linked together. Now, when we have glycosidic linkages, we will see in the next class, we are forming now from one unit, we are going to link in another unit. In the linkage of the other unit, we are going to first of all look at whether this carbon is alpha or beta. Then we are going to determine what it is linking to, or which two carbon atoms are linked. If you link a 1 and a 4 and the first carbon atom that you are linking is in the beta form, this forms what is called a beta 1,4 linkage. Okay? So, if I were to write another beta form of this. Okay. Again, I will have to number them again. So, I have 1, 2, 3 and 4. If I have now a link between the carbon atom at 1 here and the carbon atom at 4 in the other monosaccharide. Okay, so, this is one monosaccharide and this is the other one. Now, if I link these two together, it at 1 and 4, I would have a 1,4 glycosidic linkage. Okay? And depending on whether the first monosaccharide being linked to the second one was in the alpha or beta position, I would have the numbering as, or the basically the type of glycosidic linkage as beta 1,4. Okay? We will look at this in a bit more detail when we do the next lecture. Okay. So, what are the reactions that of these glucose? These are the specific reactions that can actually go on, and we looked how we can actually have glycosidic formation. What is this glycosidic formation? If we go back to the slides here, we have the linkage of the monosaccharides. In this case, what we looked at was the formation of a possible disaccharide. But if we keep on linking in this fashion, if we have a series of say beta 1,4 linkages, then obviously I am going to form what is a polysaccharide. Okay, and we will look at the different linkages as I said in the next class. Now, one of the reactions that is extremely important when we consider aldehydes is their oxidation to carboxylic acids. In their oxidation, the sugar that has a free aldehyde is usually referred to as a reducing sugar. Okay? So, the sugar with the free aldehyde, for example, like glucose. Okay? Glucose is a sugar that has a free aldehyde. So, all aldoses, what are aldoses? Aldoses are sugars with free aldehyde groups. So, all these aldoses are reducing sugars, and what happens is actually the name, the O's, when you have the name, the suffix OSE, it refers to as a sugar. Okay, so, whether it is a ketose or an aldose, it has the OSE. Okay, an aldose is where you would have the sugar in the aldehyde form. A ketose is where you would have it in the ketone form. So, the name basically is made by changing the O's ending, that is the O's, the end of the O's to onate. So, if we had glucose, it will become gluconate. Okay. Now, the test for reducing sugars is something called Felling's reaction. What happens here is you have a reduction of copper 2 to copper 1. Okay. Now, the basic reaction, this is a reaction for reducing sugars for aldehydes. What happens in that case is this is Felling's reaction. It is a very common reaction that is used in the determination of whether you have glucose. For example, it can be quantitated even. When you have a say a pathological test or a biochemical test to figure out the, in this case we are looking at glucose, but it works for any reducing sugar. Okay. So, a reducing sugar would be one that would have an aldehyde to it. Okay. So, what would be an aldehyde with a sugar? An aldose. In this case we are looking at glucose, which is an aldohexose. Okay. So, that is basically all the nomenclature that we would know for a monosaccharide. What is basically happening here is in Felling's reaction, you are going from Cu2 to Cu1. What happens is you have RCHO plus 2Cu2 plus 5OH minus. So, it is basically an alkaline medium where this goes to RCOO minus. So, what have we formed from glucose? We have formed gluconate plus we have to Cu2O. Then what happens here is this actually gives you a red coloration to your experiment. So, what we have in this case here is this is what happens in beta D glucose. Now, you recognize this as beta D glucose. So, any structure given to you, you should be recognizable in terms of where the OH is, where the CH2OH is, whether it is a fructose or rather whether it is a furanose or a pyranose. Okay? So, we have our linear form, we have our cyclized form, and the Cu2 plus, I think in my reaction I wrote it wrong, right? It's supposed to be here. This is what it's supposed to be because we have a reduction, right? So, the Cu2 is going to the Cu1. So, it is 2Cu2 plus, not here. Okay? So, we have RCH, let us just write that once more. So, for the Felling's reaction, we have RCHO, that aldehyde, plus, because this has to be reduced, going to Cu1. The first case we did not do that, 5OH minus going to, so what are we going to have here? Oxidation Cu2O plus 3H2O, and this red coloration is going to be an indicator of the presence of glucose, okay, or the presence of a reducing sugar. So, we have our beta D glucose, the D glucose in the linear form forming the gluconate, right? Okay. So, this is basically what we did today. We consider the monosaccharides, okay. The monosaccharides are the simplest carbohydrates, and we found out that they all are formed from glyceraldehyde, okay. The basic form is glycerol, then from glycerol we form glyceraldehyde, and we know that we have two forms of glyceraldehyde, D-glyceraldehyde and L-glyceraldehyde. What determines whether it is D or L? It is the disposition of the OH at the chiral carbon. If it is on the left, it is L-glyceraldehyde. If it is on the right, it is D-glyceraldehyde. Then we have the aldehydes or ketones that would have two or more hydroxyl groups attached to it. Basically, that would be the carbohydrates, and the empirical formula is CH2O whole N, where N is the number of carbon atoms present. We have aldoses and we have ketoses, depending on whether we have the oxidized carbon in the aldehyde form or in the ketone form. The open chain forms of glucose and fructose cyclize into rings. We looked at that also, where the aldehyde can react with an alcohol to form an intramolecular hemiacetal. We looked at a reaction where it is possible for the aldehyde and the alcohol to form an hemiacetal, but in this case we have the aldehyde and the alcohol both in the same molecule. Since they are both in the same molecule, we form an intramolecular hemiacetal, and then a ketone can react with an alcohol to form an intramolecular hemiketal. Okay? So, when we form these structures, we form either a pyranose depending on whether we have a 6-membered ring, or we form a furanose if we have a 5-membered ring. For the fructose case, we will have a furanose, and for the glucose case, we have a pyranose. Then we also looked at how the pyranose ring can actually adopt either a chair conformation or a boat conformation, and the furanose actually adopts what is called an envelope conformation. What is this envelope conformation? I am sure all of you are aware of this. When we have the furanose, it looks like this, right? When we have the pyranose, it looks like this, right? So, this would be a pyranose and this would be a furanose. So, what happens in this case is, it is these parts that are level, and in this case we have either the chair or the boat conformation, but in this case we have what is known as an envelope conformation. What is the envelope conformation? The oxygen is slightly lifted up from the rest of the carbon atoms, like an envelope, like a flap of an envelope. Okay? So, like the flap of an envelope, this would adopt an envelope conformation, this would adopt a chair or boat conformation. But what did we find out? We know that it is going to adopt a chair conformation. Why? Because the boat conformation would give it some steric hindrances. Okay? And then we also found out that the beta conformation would be more stable for glucose. Then we looked at a certain reaction that could go on, or the different reactions that glucose can actually have. Thank you.